All right. My job is to talk about the assignment was looking at analysis of targeted data set using a program called MetaboAnalyst. I've changed it a little bit because I actually started looking at what other people were talking about this afternoon, and it turns out that I was overlapping with things that are going on today and tomorrow. So I've changed it a little bit to mining targeted data. And so I'm going to go through, and it's actually using this the study that I showed you earlier to show you how we go about looking at integrating the data with other data that we have. Okay, so this is from Barbara Morell's paper last year, sort of looking at the workflow models for when you actually get biological data. Look at just one thing, and then you build and build, and it's really a recursive process when you look at these larger data sets. And I think it's actually important to realize that you can go back and I think I said earlier, we started this experiment about 8 years ago, and we've been mining this data. Every new platform that ever comes online, we run the same samples through, although we're not going to be able to do that too much anymore because the blood is running out. But every time we look at it, there's something new because once you learn something from another system, you can go back and take a look at this data and actually can find some really cool stuff in there. And I think I'll show you some of that. Now, there's a lot of ways to look at data online. I don't know if Al is going to talk a lot about this kind of stuff, but anyways, just to remind you that there's a bunch of different programs out there, and some are pretty good, some are not that good, some are more useful than others. And MetaboAnalyst is actually one of the better ones that's out there. It has a lot of embedded statistical data organizing, statistical management, and visualization tools embedded. And it gives you a nice first level. It actually has some pathway mapping in there now. They keep adding it. So this is really a good group, and they've really done a good job. So I would explore it a little bit. I'm going to show you a little bit of data that we dragged out of that, but mostly I'll show you it a different way. Okay. So remember, the study is to create an unambiguous biomarker of short-term and long-term feeding. We did this isocaloric feeding from a heterogeneous group of individuals at baseline. So these people were eating different diets coming into the study. Then they got all put on this polyunsaturated fatty acid diet, and then they were switched immediately to a high carbohydrate diet. The mistake, and I forgot to mention this earlier, the mistake we made when we did this study is that we did not ascertain what the diet was that the individuals were eating at baseline. Just never thought about it. But now it'd be a really good thing. In fact, there's a couple of graduate students whose careers are going to depend upon getting good dietary information and trying to link it up to really do what we're trying to do is to get this biomarker for dietary intake. All right, so this is a screenshot. I'll show you a couple screenshots of MetaboAnalyst. For the amino acids. So remember, we ran, I think, 26 amino acids that come out of a targeted amino acid profile. And we looked at baseline samples, and then there are 21 days of the carbohydrate or polyunsaturated fat diet. And if you look, using a PCA plot, basically these things don't separate. And so when we looked at the amino acid data, even though things go up and down, and if you actually do some subtraction, if you actually look at individual changes, there are small changes of amino acids, but they tend to be transient, and they don't tend to be very consistent across the experiment. Now, if you think about how we did the experiment, it's actually maybe not surprising, because we used the same amount of protein that's coming into the diet, held that constant throughout the experiment. And so in some future studies, obviously what we want to do is increase or decrease the amount of amino acids that are in the diet to see if you can actually change things. And I'll show you that there's actually some hints even within this study that this might actually work. So the amino acids, ornithine was one of the amino acids. Oh, this is amino acids. This is just rotating it and showing it that there's a lot of overlap between the baseline after 21 days on each diet. Ornithine was one. So this is another screenshot of MetaboAnalyst. It actually will do an ANOVA for you. If you have 3 different groups, it'll actually do the calculations for you, and it'll show you which ones are actually bouncing up to be a little bit statistically significant, or which will be statistically significant. And you can see that there's some of these values are just a little bit over things. And so this spot is ornithine. This is I forget. I'll come to me in just a second. Amino isobutyric acid. And so it's a minor amino acid. It's probably related to bone turnover. Why that's up a little bit, it actually is up a little bit in the carbohydrate diet. So the other compounds that we ran as the target metabolomics, acylcarnitines, which I think I mentioned earlier, are intermediates in the oxidation of fatty acids. Fatty acids come into the mitochondria and are trapped as acyl-CoAs. That's what goes through the enzyme, the trifunctional protein dehydrogenases. If you stutter, if it stops a little bit, or just you're overwhelming the system with increased lipids, these come out into the blood. Now these actually do separate nicely. So the carbohydrates, the blue and the green, separates really nicely on this. And you can actually see that the baseline people, at baseline, most of the people are sort of clustering with the carbohydrates. And in fact, when we look at a lot of the analysis we've done with these individuals, the people at baseline are eating more carbohydrates than fats. And it turns out that the non-alcoholic fatty liver disease people actually were eating even more carbohydrates if you actually start taking a look at it. So this then becomes an interesting concept about, or interesting in the way that this might be able to be used to differentiate diets. And I showed you why the 10-1 was probably one of those that drives this separation. There's actually a number of different acylcarnitines. This is a look, it's like I can't see there and I can't see here. It's like I can't see anything. This was, I forget exactly what this one was, but this is the principal component for the PSLD. So these are the loading scores. So this is the other thing that this will do. When you do see a separation in the partial least squares or the principal component analysis, it'll give you the lipids. In this case, the acylcarnitines actually drive in different things, and you can click on it. And when you click on one of these, then you'll actually get, and you'll see 10-1 is the one that's differentiating. And it gives you nice bar graphs. You can actually see these things, exactly what you're doing. And this allows you to download these things as groups and things like that. So it's really a nice little tool to be able to rapidly go back and forth and interact with your data and sort of mine it, as it were. This is a thing that Barb Morell will talk about a little bit later. This is an earlier version of a tool that was developed at the University of Michigan by a graduate student that we worked with, Gang Su in Bang Fan's lab. And this is just a heat map of the acylcarnitine. So these are individuals. This is baseline visit. This is the 4-day visit. This is the 7-day visit. These are non-alcoholic fatty liver disease people at 1, 4, and 7 days, or excuse me, visit 1, 4, and 7. So this is after PUFA and carbohydrate, baseline, PUFA, carbohydrates. And if you actually start looking up and down, you can actually start seeing the patterns of how these things change. So you can see there's a bunch of red here, there's a bunch of red here, and that red goes away in the carbohydrate feeding. And so these things go up and down. It goes up and down in a relatively predictable pattern, and so you're starting to do pattern recognition. And as Barbara has taught me very well, that human beings are visual people, and you can actually see things that you don't. If you just read a bunch of numbers, and you can do statistics, and people like George can actually do this kind of thing and actually can think through numbers. I need to see things and look at patterns in there, and it's been really helpful when you do that. And you can see down here with the NAFLD people, they really get green when they go into the carbohydrate diet. And I showed you before that the relationships between a lot of things are really robustly changed in individuals with fatty liver disease. there anything else I want to say about it? So this is the lipidomics. This is not the isocarotines, but the fatty acids. And again, remember I told you that this was, how we looked at these data, trying to find out where these different lipids changed and at what time period in the change in the feeding paradigm. And again, you can see there's differences, the different lipid classes that we see in the blood. In the principal component analysis we did. And when we do the principal component analysis with the that lipid data, you can get a clear separation of the groups, as you would expect, because that's actually, if you put all the targeted data together, it's actually the isocarotines and these lipids that really drive the differences between the group. This is some untargeted profiling that was done. Just real briefly, these are the named compounds in the untargeted, and this is partial least squares. And you can see, again, you're getting some separation, although this is not a really great separation that we get with the untargeted platform. It's unclear exactly why it doesn't change as much. These tend to be much more polar compounds, at least for the first part of the chromatogram. There's a little bit of separation in partial least squares, but it really doesn't, there's not a lot of things that drive this reproducibly. So that's how you can use these tools. So again, MetaboAnalyst is great. Click on this and it'll do this. Or click on that, it'll do that. You can flip through these different windows, and it actually allows you to do things. So I use these interactive maps a lot, and this is, again, I told you that Ala's actually trying to do this for real. But I like looking at these things because, again, you can actually see the patterns, and it can actually start giving you insights into different parts of biology. And so, again, this is these are the lipids and the saturated, unsaturated, saturated, unsaturated, and we talked about using ratios and how you can find it, and how the pattern from a baseline to a weight loss, where you've really unloaded the system. Once you're losing weight, there's not as many nutrients coming into the system. You're not overloading all the pathways, mitochondrial pathways and different pathways with nutrients. And so then starting to dig through and seeing that, well, this pattern this one catches your eyes. And what I actually suggest people to do, and it's been very helpful to me, is actually start blocking things off and looking at this. And so now you can actually start seeing. So look at the pattern here and the pattern here, and this negative relationship between some of these lipids and some of the isocarotines. And those kind of patterns change as well. And so you can start looking and you then take out your textbook and understand why these differences may be. And there's real biology in here. Okay? And if you, one of the things that I wish the Nuku map would do, but this was a feature. You could actually take both of these heat maps and actually have the computer automatically subtract them. So it would go back, take the data, and actually subtract one value from another. The equivalent value, you can take the after weight loss minus the before weight loss, you can actually see. And then what happens is that some of these things really start popping out as far as differences. And then you can start looking at the correlations between weight loss and the changes in different metabolites. And so what metabolites are weight loss. And then you can come back, and we actually now know who successfully loses weight after 2 years. And now we can actually start correlating what the metabolome is here, now what happens 18 months later, because it's a 6-month time point. And so now you can actually see, well, maybe some of these metabolites are making people more hungry, and that's why they regain weight. And indeed, these are the kinds of experiments we're trying to understand, use to look at the biological implications of different interventions. So this is a fatty acids, isocarotines, and things like that. With Jeff Horowitz, we actually took a data set. He had some people that he overfed. So this is a lean, young population, and he overfed them 2,000 calories a day for a month, which was a heroic thing. And they would only allow these people to get out of bed for an hour a day, trying to fatten them up. And really, and the experiment is to really look at the effect of growth hormone and IGF-1 and all this kind of stuff. But I said, well, what a perfect counterpart to the weight loss study. And indeed, what you can see is that you can see this modeling here, which was more akin to that middle part here. You get modeling here, and that starts to go away. Even these young, healthy people, when you start overloading their mitochondria, you can actually start seeing changes. And then you start, blind your eye to some things. And there's some really interesting things going on in these areas as well. And it's these relationships between these different lipids. And we're actually now understanding how these lipids, where they come from and how they're enriched in different particles and what they might mean from a biological process. So when you do an experiment, always think about your friends doing the same experiment, the opposite. So, we're doing weight loss, but, Jeff's overfeeding people. And so looking at this and looking at the yin-yang. So somebody asked, Taiwan asked, what about, can you tell if somebody's been fasting or if they're in negative energy balance? That's the other thing that we'd really like to do, to see if there's a way you can actually see if a person is actually losing weight or gaining weight. Now, we can, you can tell if somebody's losing weight if they're actually losing weight. But when you get a big cross-sectional study, can actually see who's actually in negative energy balance or positive energy balance. Are they on their way up or are they on their way down? And is that predictive for other disease states, atherosclerosis, diabetes, those kinds of things? Okay, so so these these are big differences, and they probably tell something. Now, the unfortunate thing, if I knew what I know now, then I would have had Jeff draw blood, after the third day and after the sixth day, so we could actually see what the acute changes of overfeeding are versus the chronic changes. And so how long does this take? Because we've done some weight loss studies, weight loss changes, you can see that when you change diet, things change pretty quickly. When you start losing weight, things change very quickly in the metabolome. And so this negative weight loss can make a big difference. And so when you're thinking about, your cancer patient who just got radiation therapy and they are not feeling well, and you take their metabolome trying to predict their outcome, you got to remember that even that day, those couple of days, they're really going to affect the metabolome. And so if you're going to do sort of control groups, you need to think about the physiological status, especially if you're doing human studies. Obviously critters are a little bit easier to pair feed and things like that. All right. So one of the things that we did is we fed everybody these diets. Now, there's a range of carbohydrates and a range of lipids in these diets, and so looking over those, looking over the dietary records. So what we did is actually had all the diets that we had prepared for people and gave those diets to independent or blinded dietitians, and they actually came up with what they thought was the caloric intake from different macronutrients. So basically doing a dietary recall in a controlled manner to see how closely it actually matched up. And so they actually did pretty well. Of course, people are in a study and they make it. But I'm just going to point out a couple things. So if you look at the fatty acids in the blood that we measure and the percent calories that came from carbohydrates, so this is this cool map program. Again, you can cluster things, and within this cluster, sorry, So this here is the percent calories from carbohydrates and percent of consumption of carbohydrates. And within that cluster are all these saturated fatty acids. And so remember before I said that that what we see is when you feed the high carbohydrate diet, you actually see an increase in saturated and monounsaturated fatty acids. Within this cluster now, independently, we got the amount that was eaten by these individuals, and we took the metabolomics data and we put it in together with the dietary data, just put it all together, let the computer find the relationships, and they actually cluster together. That's really satisfying because it actually says that maybe this is actually going to work. If you look at amino acids and percent calories from proteins, so remember the proteins weren't really differentiating people very well from different diets, but if you actually look at the percent calories from protein and you look at this cluster, all the damn amino acids are in there. That's, again, really kind of cool. So it really does tell us that the relationship between dietary intake and what you measure in the blood, it's potentially possible to really get an idea, both how much they're eating and what kinds of foods they're eating. So we're really excited about this, and hopefully Jim is going to make this all work, right, Jim? He's going to be Dr. Jim. He's going to know it all. Anyways, all right. So is Karen here? Oh, too bad. I wanted her to see that. All right, so lipidomics. So TM's going to tell us about lipidomics. I don't think Tanu has been doing all this. This is some data that we looked at. We did a large-scale lipidomics study, and this is what we're really going to be hopeful that this is actually going to give us this way of really separating these diets and perhaps the amount of intake, of variable intake. And so this is looking at carbohydrate and PUFA diets. And different symbols for day 2. So visit 1 was baseline, 2, 7, 21 days, 2, 7, 21 days of the carbohydrates or a PUFA diet, and here's the plot. Couple things to notice about this is that things separate very rapidly. So all the blues and the greens actually are separated. There's a little bit of muddling up in here, and it really is part of the early to day 2 time point, but they're very rapidly separating. So remember, these are paired individuals, and so we actually can connect each one of these spots to each one of these spots. The baseline, again, is clustered, and more of the baseline is clustered with the carbohydrate diet. And then there's a cluster right here. You see all these little ones right here. These are the pooled samples that we put together from all the samples we put in, and which is very satisfying. And when all these things get together in one place, it means that you know, throughout the runs when we actually did throughout the run, when we actually assayed these samples, even throughout the run, they cluster together, which you know, tells us that the experiment is working the way it should. So you can really do that. So this is the kind of way that you can do it. Once you start putting your data out there, you can actually start looking at the temporal relationships, even when you're doing a lot of time points and different interventions. And then this is using this cool map application again, but you know, the lipidomics, there's lots of data and it's really good data. This is a really fine platform. And you know, and so this is just looking at this same the PCA plot differently, but it's just, so this is a radar, and we're looking at this little piece. So this is how much data is there, and I'm just focusing on one because it's a pretty picture. You know, sort of your Christmas colors. One of the things I want to point out is that you can rapidly see here. So this is baseline, visit 1, visit 2, baseline, sorry, after 21 days of PUFA, after 21 days of carbohydrate diet, and then the people with non-alcoholic fatty liver disease are down here. What's interesting is that there's I want to point out 2 little ones here. This one and this one are the same person with 2 different, the same person. We actually think the samples are mixed up. And we've been, and if you look at the tube that it came out of, 4 and 7 are the numbers that are on there. You know, you can't really tell. And so, you know, what do you do with this? Well, I mean, look at this green. It's all red there, and all green here and red there. You know, my guess is that we switched the sample. And so you can actually use this to even vet your data to really understand perhaps there's things going on that are even from a technical standpoint. So then the question is, what do you do with that sample? You just throw it out? You probably have to. And finally, somebody asked a question about fasting in critters. This is a rat study, 24-hour fast in a rat. And here are the metabolites, so just put it into the circulation, the metabolites that change with 24-hour fast from an ad lib fed animal. And you can actually see that there's a number of things that all make sense. You know, your butyric acid, so a ketone body goes up, you know, the lipids go up because you're mobilizing fatty acids to feed the animal, your amino acids go up because you're fasting and you're doing gluconeogenesis, you're breaking down protein in the muscle, so everything makes sense. So these are the kinds of things that you can use to actually start looking at how long animals are fasting or how long humans are fasting and things like that. So, so those kinds of things, and then starting to take ratios of different things that go down, because there are stuff that goes down. You know, these are, you know, up 60%, but there's actually some that go down as well. And so if you actually start taking these things, maybe you can actually start teasing out different aspects of physiology. So I'm going to stop there. This is the way we look at the targeted data and a little bit of broader data with this, and just thinking about how to look at the data and actually putting it together with the other types of data. Now, somebody asked earlier, can you do this with transcriptomic data? Absolutely. You can do this with anything. The thing we like to do is clinical data and looking at the relationships of different clinical data. Some of the stuff in there, if you look at it, there's some interesting relationships between blood pressure and different amino acids and things like that. So there's things to dig out, how this all is going to sort out and how you're going to make physiological messages out of it isn't quite clear, but it does seem to work. Questions? You look like you're ready.
Don't you think you need some genetic data as well to predict your responses, like SNP the most important genes? Or stuff like
it's a
this?
great question. So Bob Gertson was here a couple of weeks ago from Harvard, and he's actually done 130 metabolites or whatever he does with this thing, and actually in the a couple thousand samples from the Framingham Heart Study, they have GWAS data, and he's using these as MQTLs. I showed you earlier that we have this animal model where we actually bred for high and low running capacity. And they clearly metabolize different substrates differently. We made a 700 animal F2 cross, and we're actually doing the metabolomics on all these, if I can find the money. At least we're going to do a subset and actually use that to see if you can actually use it as basically an MQTL, so a metabolite quantitative trait. Gerson states that he believes that the metabolites are lining up with things that either generate or consume those metabolites. Not a lot of them are actually reaching real high statistical significance. George and I are on a grant with Sarah Tishkoff, who has the DNA samples that she's run from about 1,800, something like that, people from different tribes in Northwest Africa, Northeast Africa, excuse me, and they're either hunter-gatherers or they're pastoralists and things like that, and they eat widely different diets. And so we're going to take and see if we can actually see with this dietary, the immense amount of genetic heterogeneity even among tribal people in Africa, and see if we can actually use that to do exactly what you've said. So, yeah, it can be done. Well, so it can be done. Is it going to be successful? That'll be the question. Yes, sir.
So what are the difference between the lipidomics assay and the metabolomics assay?
So I mean, lipidomics is sort of a subset of the untargeted metabolomics platform. So the biggest difference is that it identifies specific subclasses. So TM will tell you about exactly how it's done, but the lipidomics will give you specific subclasses and chain lengths of lipids. And you get, up to 1,000 or even more of those. There's a lot of data in there that still needs to be sort of sorted out. The untargeted platform, there are lots of lipids in there. A lot of the stuff that's coming out late in the chromatogram are clearly lipids, but they're just hard to identify because, the molecular masses of these things can be a number of different things. One of the things that we're going to start working through is actually correlate the lipidomics with the untargeted metabolomics and see exactly how these things correlate. And it's part of the project that Al is working on. Is to really try and use these different platforms to seed and to try and cross-identify the, quote, unknowns in each platform. And so if you find a very high correlation between this lipid and the untargeted and this lipid or presumptive lipid untargeted and this lipid identified over a number of samples and they co-vary in a high degree, you have a much higher likelihood, especially if the masses are similar, that this is probably the same thing. And then you can do some other things to specifically identify what that unknown in the untargeted platform is. And you can do it the other way around. You can find known things in the untargeted platform and hopefully find it in the lipidomic platform, because the chromatography is different, but it's not that different that you shouldn't be able to do it. Anybody else? All right, thanks. All right, so next,